All right, so we've seen how the neuron operates. We get it excited, shoots the action potential, and then we can get communication between the neuron and whatever target cell it's talking to. So now we're going to focus on a specific branch of the nervous system. We're going to look at sensory physiology. We're going to be looking at those afferent pathways. We're going to bring information from the peripheral back towards the central nervous system, where we can get integration of what we're actually sensing. Okay. There's multiple branches of the sensory. There's the special senses, the general senses that are throughout the body, and there's also visceral sensory. The visceral sensory will focus more when we get to the various organ systems that use those sensory inputs. Example is like the baroreceptor, senses that stretch on the blood vessels, right? That brings information in. So that would be a visceral sense. And the sensory that we're going to be focusing on mainly is going to be the general senses, or senses that lie throughout the body. Specifically, we're going to look at cutaneous, so information we get from sensory information close to the skin. All right, but that lies throughout the body, so it's a general sense. And then we're going to focus on special senses. Special senses reside in the head. We got taste, we got smell, we got equilibrium and auditory, and we got visual. All right, so we'll focus on those. We'll first look at how we actually get the sensory information. How do we send it or get it transduced, is the word, into an action potential. We'll look at the general senses in this first part one of the sensory, and we'll look at smell, and we'll look at taste in this first part. The second part will solely be focusing on the ears and the eyes. The eye, visual, is a very complex sensory organ. Same with the ears, are a bit more complex. So part 2 will deal just specifically with those, the senses that come from those two organs, the equilibrium, the auditory, and the visual for part 2. All right, so let's jump in. We'll start looking at part 1. Again, we're going to start with the general and then work our down into the general senses and then into taste and into smell. All right, to make this all work, we have to have 3 main components to get an actual sense. Because what are we taking? We're taking stimulus. Stimulus can be outside the body, say like light coming into my eyes. That's a stimulus coming in. But what we're doing is we're going to basically take this information, and we have what's called sensory receptors that are going to do what we call transduce or transduction. All right, because what are we perceiving? We're perceiving stimulus. Our brain doesn't perceive, hey, here's light. What's your brain's, what's the language of your nervous system? It is action potentials. All right, so we have to take the stimulus and turn it into an action potential. And so that is a process called transduction. And that is one of the main components, is we have to have sensory receptors. Sensory receptors respond to whatever stimulus that activates them. Okay, for light, it's the photoreceptors in the eye. Okay, for smell, there are neurons with receptors on them that respond to different odor chemicals. Okay, and what do? They basically, these receptors turn those stimulus, they're activated enough into an electrical impulse, into the action potential. And that process is called transduction. Okay, there's 3 kind of main ways these neurons can be set up for sensory. And how do we get our neurons to fire? What do we learn in nervous system? We learn that our nervous system, our neurons have to be excited to get the action potential to take place. In this case, it's going to be the stimuli. Whatever stimuli is what's going to excite the neuron to cause an action potential. Okay, and we're going to see with our sensory receptors how this takes place. where would our sensory receptors, since they're the ones responding to the stimulus and then going to cause or transduce it into activation, where are we going to find them? They're going to be on the dendritic end of the neuron, right? And so we can see there's two main setups here. There's a third setup we'll talk about, but you can have a sensory neuron that basically has free dendritic ends. I know this is these are dendrites here. Okay, this would be where the sensory. These are just open nerves. There's nothing special. They're just dendritic ends. Okay, so we can have what's called free nerve endings. That would be this setup here. All right, our dendritic end can have a specialized structure, like here. A modified dendritic end. If you remember your Pacinian corpuscles from your anatomy days, we have the specialized modified dendritic end, and here is the dendritic portion of the sensory nerve. Okay, so we can have free nerve endings, we can have modified dendritic ends. There's also receptors that are actually not even neurons. They are usually specialized epithelial cells, all right, that respond to the stimulus. But these specialized epithelials have to be associated with these sensory neurons, because we got to get the transduction of the signal and then send that signal up into the central nervous system, or into the central nervous system. Okay, so three main setups: free nerve endings, most basic, it's just nerve ending where we have the receptors. We can have a dendritic end with modified into it, like we see here, and then we can have more complex where we have these specialized epithelials that transduce the stimulus signal. And you see them, they operate very similar to these specialized epithelial, operate very similar to nervous tissue. Voltage gated calcium channels, transmitters, so they have vesicles. When activated, they'll release neurotransmitter to the sensory neuron. Okay, and cause activation, right? But it is the stimulus. The transduction is what's going to give us that excitatory input, okay, to get our neuron activated to elicit the action potential. Okay, but to make this all work, we have to have three main components. We got to have the receptors. You got to have the receptors that are operating properly so they can transduce the signal. Then you got to, once you transduce it and the neuron gets excited, we got to have the sensory neurons that can take it to the region of the central nervous system where we can get integration, where we can put perception. So you have to have that region of the brain operating to get sensory, to get the senses. Okay, if you remember your anatomy days, we got our somatosensory cortex. Those are going to be our cutaneous information is going to be going up to that part of the region, right? Our special senses all have their own little cortexes. Okay, olfactory for smell, auditory, okay, visual. But again, I want to make the point that all three of these have to work. If your receptors are damaged, you listen to very loud music and you damage your little nice sensory receptor cells in your ears, all right, you're going to have some hearing loss. Or say you get hit somewhere in the brain where it damages those sensory neurons. Now we may be able to transduce it, but we can't get the information back to where it needs to go in the central nervous system. Right? And then we have to have the region of the brain operating that to put the perception to. So if you got hit and some reason you had a hematoma that basically killed a lot of these cells here in the olfactory cortex, this may work, the sensory neurons may be carrying it back, but if you don't have the neurons to do the integration, you will not have that sense of smell. Okay, so you got to have all three working to make this function properly. So since our sensory pathways start with sensory receptors, let's take a look and let's classify and look at their functionality, and we can classify them as by their modality. What stimulates the receptors? What's going to cause them to be activated to ultimately, if excited enough, to cause that action potential? Right, and the first, chemoreceptors, you imagine what stimulates them are certain chemicals. Right, taste. You eat foods and they have certain chemicals in them. Those are going to activate receptors in your taste buds. Smell. All what you're smelling in, or you're smelling in these odor molecules. The odor molecules are going to activate receptors, chemoreceptors in the nose, and that's going to elicit a response. Okay. The broader category, mechanoreceptors. This usually is distortions to the membrane. What can cause distortions? Stuff like touch, vibrations from hearing and sound, acceleration with equilibrium. Okay, those are all can distort the membranes, and that's what mechanoreceptors will respond to. Photoreceptors, you will find those in the eye, and what are they going to respond to? That light waves or photon energy are going to activate the receptors, the photoreceptors. And then we have chemoreceptors, or sorry, not chemoreceptors, thermoreceptors. Thermal, in that they respond to cold and, or warm and cold. There's another class 2 I mentioned here, but the stimulus form is multi-dimensional. There can be many different stimuli that can activate these, but it's kind of the one we don't like to talk about. So that class of receptors are that respond. They can have many different types of stimuli, at least in modality-wise, are those nociceptors. Okay, they can be polarized to, say, extreme heat, extreme cold. So kind of similar to thermoreceptors that are responding to temperatures, but these are on the extreme end. Extreme pressure, like when you get hit, that has lots of pressure, and chemicals. When tissue is damaged, many times chemicals are released, and so those nociceptors can respond to various chemicals. So you can see they can have mechano modality, they can have a thermal modality, and a chemical modality. Two of the main neurotransmitters in this pathway is the glutamate and substance P, okay, along with, we'll see the opioids. All right, endogenous opioids in this place. You actually have opioids, neurotransmitters in your system. The ones we think about are the drugs that elicit the activity at the receptors for these endogenous opioids. Okay. So our receptors can adapt, and this is important. So not that we're in class anymore, but if you're sitting in your chair looking at your computer studying, you don't want to be thinking about, hey, there's pressure on the back of my leg from the chair. When you're studying, you want to think about what's on the computer, what you're listening to Professor Huss and all his glorious knowledge he has, and you're focused on that. You're not focused on the pressure, because the pressure between that's on your legs from the chair is still there. It's just you're not making yourself aware. And so receptors can have what we call sensory adaptation. Okay, they adapt so you can focus on things that are essential and not have to focus on things that are non-essential at the time. Okay, so sensory adaptation keeps you from stop being aware of, say, a continuous stimulus. You're still sitting on the chair, you're still having pressure on the leg. Those receptors are still getting stimulated, but you're not consciously thinking about it because they are phasic receptors. They are fast adapting. All right, and this is what happens with these receptors. They will adapt, and what they do is, you can see here, if this is that sensory neuron, all right, here are the action potentials elicited by the receptors. But if the receptors adapt, right, when you first get the stimulus, boom, boom, firing, your brain's making notice, hey, I'm getting rapid firing from those pressure receptors on my legs. All right, I'm feeling pressure. But as the receptors adapt, the rate of firing of that sensory neuron in the brain is reduced. Okay, there is less firing, and now what signal is the brain getting? The brain can only put perceptions to different frequencies of action potentials. Okay, remember, coding is in the frequency of action potentials. Hey, I'm not getting a signal anymore. All right, don't need to focus on that. Okay, so those receptors, those phasic receptors, the fast-acting receptors, slow down the frequency of firing, and now I don't have to continually think about this. Okay, if anybody's gone into a house where there's a gas leak, they put a certain chemical in the gas to give you that smell. When you walk in, you smell it right away. All right, but if you stay in that house for a while, you stop recognizing that odor because your receptors, in this case, you kind of didn't want it to adapt because you do want a gas leak, you want to know it's there. But if you walked back out of the house, let your receptors readapt and came back in, you would smell that odor in the gas again. Okay, but phasic receptors are fast adapting. There are receptors that are called tonic receptors. So they maintain their firing. They don't do this adaptation. Or if they do, it is very slow. It is not very fast adapting. And you see when the stimulus is applied, boom, 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 it's still there. All right, we still have a higher frequency. And where might this be important? What kind of signal would we want a tonic signal for? Right? How about something like pain? Right? Fortunately, some pain like joint pain or whatever, we want to go away, but it means there's something wrong. If you put your finger on a hot stove and your pain receptors that respond to that extreme heat are phasic, you could, as long as you kept it there long enough, all right, you'd stop feeling the pain. But luckily, these are tonic receptors, and you're going to continually feel that pain because you, oh, I want to get it off. Okay, so pain receptors, tonic, not fast adapting. Okay, where stuff, phasic receptors are those that are fast adapting and let you filter out unimportant stuff so you can do stuff like study, listen to your professor on the computer, expounding his knowledge. All right, but phasic receptors and tonic, not adapting, fast adapting are the phasic. So let's elicit, because what we're doing with these receptors are we are taking a stimulus and activates the receptors to elicit a response. If there's a strong enough response, there's a threshold response, we will get an action potential. And that action potential, as long as all the sensory neurons are fine and the brain is fine, we can get that information out to the brain where it needs to get integration. Okay, so how do we do this? If you remember our neurotransmitter and our nervous system, we'd have ligand-gated, say, ion channels that would elicit excitatory response. Instead, our stimulus is eliciting excitatory response in our receptors to stimulate our sensory neuron. Okay, these depolarizations elicited by stimuli are called generator potentials. All right, similar to what we say, excitatory postsynaptic potentials. Okay. So we have to get enough of the stimulus to get us the threshold to be able to elicit the response. Because I could be actually touching my skin, but if it's not a strong, I'm not activating that receptor strong enough, it is sub-threshold, right? I'm not going to get a conscious awareness of that. Okay, it's got to be strong enough to where I actually feel the response, because that strength, strong enough signal or strong enough generator potential will get us the threshold, and now I will send the information from the receptor through the sensory neurons up to the brain. Okay, so I can have touch going on, but if the generator potential is below threshold, I'm not going to have a conscious awareness of that taking place. Okay, so it is just like our graded potentials, our receptor or generator potentials are a graded response. Okay, very, very, very light touch, a little bit thicker, a little bit harder touch, not much touch, or more touch, and now we get threshold, and I can actually have an awareness of the touch taking place. Now, say I get to threshold, I have a response, but I'm just above threshold. I'm at negative 54 millivolts. Okay, just a small amount of touch, but I did reach threshold, so that information is going to get sent to the brain, and I'm going to have a conscious awareness of that thought. But what happens when there's lots of pressure on that receptor, a big strong stimulus? How does my brain get an interpretation of that? If you remember, how does the nervous system code for stimulus strength? Because the action potentials, whether I reach negative 55 or whether I reach positive 10, right? My action potentials are the same strength. And so how do we code? All right, it is by frequency of action potential. Say here's our receptor here. I'm getting this weak stimulus, weak pressure on here. Okay, but it's above threshold, so I'm sending action potentials, but the frequency for that weak signal is much slower than the frequency of signaling that I would get with a strong stimulus. Okay, so larger stimulus is going to result in higher frequency signals that gets sent to the brain, and the brain, those integrating areas of the brain, hey, I'm getting this low frequency, I'm getting high frequency. Your brain can put an interpretation to that frequency of signal. All right, and that's how you get the perception. Hey, it's just a small amount of touch versus it's a very strong amount of touch. So our receptors are very specific for certain stimuli. Okay, the stimulus in which they are most responsive to is called the adequate stimulus. So say for a photoreceptor, that can be photons of light. So it'd be very, very sensitive to photo energy. Okay, that would be its adequate stimulus, what it responds mostly to. Okay, some of these receptors can respond to other modalities, but typically that modality, if it's another modality, would have to have a very, very strong stimulus. An example of that is our photoreceptors. Okay, our photoreceptors respond to light. You only need a small amount of light to actually elicit a response in the photoreceptors. But if you had a hard blow to the head, that actually will elicit some of the photoreceptors. That's the response and where you're seeing stars, see things, because it does elicit a response in the photoreceptors. Okay, but it's not its adequate stimulus. Adequate stimulus is what these receptors mostly respond to, or respond most readily to, right? For chemoreceptors, okay, taste, smell, pain, all right, chemicals dissolved in saliva, and the mucus for smell, and pain, the extracellular fluid. Okay, warm and cold, increase in temperature, okay, and mechanoreceptors, you can see they have different adequate stimuluses. Okay, where the Pacinian corpuscles respond to pressure, where hair cells respond to sound waves. Okay. So cutaneous senses, the ones we'll focus on here, are throughout the body. They are the ones that are detected near the skin, okay, perceived by the brain. They are the somatosensory sensation, and you're going to see they go to the somatosensory cortex in the brain. But these cutaneous, we have mechanoreceptors, okay, respond to touch, vibration, our Pacinian corpuscles, Rufian endings, Meissner corpuscles. These respond to deep pressure, light pressure, vibration, okay, because those stimuli are going to distort the membranes of these receptors to get a response. Thermoreceptors, okay, we respond. If you put coal, you jump in the ocean, you feel the cold because those thermoreceptors on your skin are responding to that change in temperature. And we'll see there's a range within these. Warm receptors 25 to 47, 12 to 35. You can see there is an overlap. So there's a region in which these receptors might be responding equally between that 25 and 35. Not equally, but they have some overlap in there. Okay. Imagine stuff above here, above 47 or below 12, right? Those don't elicit a response in the cold receptors per se, but what are they going to elicit the response in? They're going to elicit the response in nociceptors. Okay, nociceptors respond to extreme cold, like below this range, extreme heat above this range. All right, tissue damage, pH changes, any inflammatory response. Remember, those nociceptors have multiple modalities in which they respond to. So here are looking at the cutaneous receptors, those that lie within the skin here, or lower, just below the skin. Okay, we have free nerve endings. Okay, that houses stuff like light touch, which would be a mechanoreceptor, hot, cold. Okay, our thermoreceptors and nociception, pain. Right, we have Merkel discs. Okay, we have Rufian corpuscles. Okay, you see the Rufian corpuscles here. Okay, deep in the dermis, stretch of the skin would be an elicited response. They would stretch the skin, cause a distortion of the membrane of these Rufian endings, and that would elicit a response for signaling. Our Meissner corpuscles, you see here, where are they located? They're located here, not very deep. Okay, slow vibrations, change in texture. They're close to the skin, so they can detect changes in texture. Okay, where we wouldn't want to elicit our Pacinian corpuscles, okay, because they are deep here. They couldn't listen. We'd have to have a large change in texture to elicit a response in a Pacinian corpuscle. So you can tell What's the function of these? Depends on location and structure of them. They're going to respond, sitting in, they're going to respond to stretch, but they are going to respond to deeper pressure. Where Meissner's, again, are mechanoreceptors, but they are elicited just underneath, or sorry, just under the dermis there, or the epidermis, and they can respond to smaller changes in, say, texture, so forth. Doesn't have to be deep pressure. Okay. Here's a closer look at the cold receptors. Those are the free nerve ending. One receptor type I like you to recognize here are these transient receptor potential channels. All right, they are channels for ions, for sodium and for calcium. So eliciting a response on these receptors allows for sodium and calcium to move in. That depolarizes cell, and if it's a strong enough signal, we get enough depolarization through these transient receptors. All right, we can get a receptor or generator potential. That is threshold, and then the action or the sensory neuron will fire. Okay, these cold receptors. The opening of these channels happens in response for cold receptors and stimulated by the range of the cold. Okay, and you can see there are several types. So some of the cold receptors are at the lower end, where there are other transient receptor potentials that respond to temperatures that are in the higher range of the cold. Okay, but then when we get higher than this 25, they begin to be inhibited, less opening and less response through them, and therefore less likely to have those neurons firing. Okay, our warm receptors and TRP. All right, you can see temperature insensitive neurons don't change with changing temperature and firing. But your warm sensitive neurons, those that have warm receptors on them, you see as we go down, this is going down 32 to 39. Again, you see as we get it to the higher temperature, you can see the frequency is much greater than when we see at the lower temperature. Okay, so the TRP, transient receptor potentials, they open to warmer temperatures. As they open, now sodium and calcium in, and that's going to cause a higher firing, more excitatory signal in the neuron, and will cause them to fire more frequently. Okay, just the opposite of what we saw with the cool. The warm receptors are inhibited when the temperatures start getting cooler. As we get into cooler ranges, this frequency will even drop even slower. Okay, maybe we'll be in that range when we start getting, say, down to 25 degrees. Okay, but they are these TRP, transient receptor potential channels, that respond open, allowing those ions to move through. Now there are hot receptors, but where do you think hot receptors fall in? They fall in the line of nociceptor. Okay, stimulus special to nociceptors. Again, there are TRP channels, but they're not going to be open when we're down in these cooler ranges. They channels only open when we are at these higher ranges of temperature. All right. So again, at the higher temperatures, that elicits the opening of the channels, and now ions will move, and those pain receptors or pain neurons will fire more frequently. As we go up in temperature, you can see they fire quicker and quicker. So once we get that transduction to place, the receptors transduce the stimulus, we get the neuron activated, and we got to bring that information up to the brain so we can get interpretation of it. And these sensory pathways are usually multi-neuron. We'll have a first order, second order, and most times a third order neuron that complete the pathway to get it to the specific region in the brain that it needs to go. All right. In the case of the cutaneous information, where in the spinal cord, because we're bringing the information into the central nervous system and we're going to have to carry it ascending up to the brain. So we have these ascending tracts that carry this information. And for cutaneous, we have for sensory and crude touch and pressure, they're going to fall through the anterior spinal thalamic pathway. Okay, you can see these pathways here are what we're talking about. We got the anterior spinal thalamic pathway, looking here, and then the lateral spinal thalamic pathway is going to carry pain and temperature information, and that's located there. So these are those neurons that are going to be, these axons that are going to be projecting up to the brain, okay, to get us to the right. In this case, since we're looking at somatosensory information, sensory information, we're going to bring the information up to the somatosensory cortex. So let's look at how this information travels, and we can see it's multi neurons. Here's the primary neuron or first order neuron. All right, we've received the information. We got the neuron. This is a neuron that gets excited from the receptor end, and we send it in to the spinal cord. Okay. Many times the information, especially in this case, it's going to be, we're actually going to cross over or desiccate. Hopefully that term looks familiar from your anatomy days, in that the left sensory information is going to go to the right side of the brain. It's going to be contralateral. Okay. Also, there's somatotopy, and we'll talk about that in a second. But let's get our information, our sensory information, up to where it needs to go in the brain. Okay. So spinal thalamic, to map this out, is you can see where do we synapse between the first and the second order neuron. All right, you can see it happens in the spine. First order, second order, okay, in the spine. Right, the second order neuron is actually going to desiccate, cross over, and then that information is going to travel up, depending on whether it's pain or whether it's crude touch and pressure. Okay, crude touch pressure is going to go to the anterior, where the pain is going to go through the lateral, but it's going to travel through those pathways all the way up to the thalamus. Okay, thalamic, where do we get the synapsing between the second and third, or what they're calling the secondary and tertiary neuron, is in the thalamus. So first one's spine, spino, second synapse is thalamus. Okay, and so we synapse the third neuron at the thalamus, and that third order neuron is going to take the information to the ending spot where we go to the primary somatosensory cortex in that postcentral gyrus of the cerebral cortex. Okay, so our somatosensory information goes up to our somatosensory cortex. Okay, so that is the path, that is the pathway in which that information will travel up to the brain, so we can get integration on what is actually being sensed. We can get perception of it. do want to make note of one of those regions we saw there, the thalamus. Thalamus is a big player in sensory information. All sensory information heads to the thalamus, and the thalamus is basically kind of the way station that's going to send to the different regions of the cortex for the different stimulus, for the different senses. Okay, if it was olfactory, or sorry, not olfactory, because olfactory is the only sense that doesn't go through the thalamus. Okay, but let's say we got optic information. It's going to go here and then up to the visual cortex. So the thalamus sends it out to the visual cortex. Or if it's somatosensory, the neurons from the thalamus are going to send it up to the somatosensory cortex. So it's kind of like a relay station for all the sensory information. It's not solely just a passive relay station. They're finding out that it does some editing of the information. But what I want you guys to realize is that the thalamus is kind of where all the sensory information comes, and then it gets pieced out to the regions of the cerebral cortex in which are devoted for those different senses. Okay. And so for our case, where did we get sent to? We got sent to the somatosensory cortex. Because all that cutaneous sensation is all going up to that somatosensory cortex. All right, there's the central sulcus there. So this is the post-central gyrus. And I mentioned somatotopy. Okay, they've mapped the somatosensory cortex, and they've mapped to show that different regions, senses from different regions, go to different portions of the somatosensory cortex. Besides being mapped, I do want you to realize is, look at the regions, some of these regions here that are mapped of the body. Look at the fingers. The fingers have a very large section of the somatosensory cortex here. That section is devoted to hands and finger. Okay, your trunk is a very large part of your body, but look at how much region is devoted to your trunk. Okay, your hands very small compared to your trunk, but for the somatosensory cortex, there is a much greater region of it devoted to the hands. So what's this probably tell you about the amount of receptors in these different regions? Yeah, we have lots of sensation in our hand compared to the reception that we have in our trunk. Okay, we have low density of receptors in our trunk, very high density in our hands, which makes sense because we basically feel the world through our hands. Okay, also remember this was contralateral, so we're on the left side of the brain, so this would be for the right portion of the body. Sensations coming from the right side are going to go to this left side of the somatosensory cortex. And depending on what region from the right side depends on what region that sensory information comes to. Okay, that is called the somatotopy. So because these pathways are multi-neurons, we can get what's called referred pain. Okay, we may feel pain in certain regions around the body, and it may be due to some sort of organ. All right, pain elicited from damage to an organ or disease in the organ or so forth. Anything that would cause pain in one of the organs possibly might cause pain in different regions of the body cutaneously. Okay, you can see the stomach sits here. Okay, you had a pain in your left arm, it's a common side of a possible heart attack or something wrong with the heart. Okay, the esophagus, even though the esophagus runs here, could have pain throughout your left chest region. And what's the reasoning behind this? In that it is believed that these sensory receptors for pain, okay, here's the region of the skin, right? Say this is the skin region, sensory neurons that are located in this left part of the arm. These pain receptors for the heart are going up and they're synapsing on a common neuron. Okay, remember the brain can only perceive the information it's getting. So if it's the heart's eliciting this neuron and it goes up to the brain, it could elicit it as per the perception as pain in this region of the skin, and therefore that's why when a person has a heart attack, they may have this referred pain here, because these neurons, pain receptors here, are activating a neuron in which that area of the skin, okay, meet up on the same neuron. So this is important for a medical sense, in that if a patient comes in with a pain in a specific region, that can, oh, I got a pain here, it can pinpoint maybe what's going on internally in various organs. So jumping back a little bit, let's look at how tactile discrimination, how do we know where stuff is being touched, how do we discriminate between things? And so one of the common things, if we were in lab, we would be measuring what's called the 2-point threshold for discrimination. Okay, ability to differentiate information detected through touch. How do I feel 2 different points? Okay, and this can vary between body regions. Okay, remember this guy down here. This was that somatotopy. Remember, our hands have lots and lots of receptors because lots of the brain region is devoted to the hand, where our trunk has very low density receptors. Okay, there's a very small part of the somatosensory cortex that's devoted to the trunk because there's not as much sensory receptors out there, not as much density. Okay, so how does this relate to be able to tactile discrimination, be able to distinguish between feeling 2 points? Okay, and so 2-point threshold is telling us what is the minimum distance that 2 points can still be perceived as 2 separate points. And so this has to do with the receptive fields for our sensory neurons. Okay, we have to have sensation. We need to have the ability to feel senses throughout our trunk, and we have to have the ability to sense it throughout all the areas of our skin. But if we have low amounts of receptors in a certain region, each receptor is going to have a larger field to be able to cover that larger area. All right, and that's what we see basically when we see like this. Okay, this would be a large receptive field for a receptor, okay, on this side. So if we were in lab, we would have you guys have calipers, and you would be having the person close their eyes, and you would touch their skin with it, and at a certain point they would tell you when they could only feel it as 1 point. And you can imagine in regions where there is low density receptors, where the receptive field has to be big for each receptor, this right here is only eliciting a response from 1 neuron. The brain's getting information, hey, this neuron's firing, and it detects it as perceives it as just 1 point. Whereas if I have smaller receptive fields, I have lots of receptors, so I can have small receptive fields, right? The same distance, I can get and elicit a response from this neuron and this neuron, and now my brain can perceive that as 2 points. Okay, so you can imagine where are areas that we have a high amount of density of receptors in our fingers. All right, so we have a very small, it's inversely related in that the more receptors, the lower the 2-point threshold, because I can determine 2 points very close together where I have lots of receptors. Where I have very little receptors, my 2-point threshold would be much greater because I could be pretty far distance out and I could still, if I have low density of receptors, perceive that just as 1 point. Okay. And so just by looking at this guy here, you can imagine the regions in which I would have low 2-point threshold and a high 2-point threshold. Right, you see there? Right, they're inverse. High density means low 2-point threshold. Low density of receptors means a large 2-point threshold. So for tactile discrimination, you can have lots of receptors. That gives you better tactile discrimination. Your body can also help fine-tune location by using what's called lateral inhibition. Okay, you can see receptors. If I put this little pin down, here's what gets elicited. Here's the strongest response, and even when I push the pin down, other regions around there will also get elicited response, but a lower response. Okay, so your sensory actually gets responses from outer regions and where exactly the pinpoint was. What lateral inhibition means is you can see this first order. So remember our sensories, 3 orders of neurons. Okay, and remember your brain can only perceive what it is receiving from the sensory. All right, these are the neurons that talk to the region of the brain to perception. These are the ones involved in sensory. Okay. And what the lateral inhibition means is, look at these second order neurons. The neuron that is strongly elicited will strongly stimulate its second order neuron, and these lower stimulated neurons are traveling in the same pathway. All right, it's the same information, but what is the second order neuron doing? It is inhibiting these neurons that were weaker in stimulus. All right, so what information does the brain get? If I inhibit these second order neurons, I'm inhibiting this neuron talking to the brain and this one talking to the brain, and I'm just getting this one talking to the somatosensory cortex. So now where's the perception going to be? It's going to be right where that pinprick was at. That allows us to discriminate and give us that finer location or tactile discrimination. So you can see that's why we call lateral inhibition. It's basically inhibiting those neurons that are lateral to it, and therefore it allows us to get this fine tooth discrimination of location of where the touch is coming from. Okay. All right, so we're through with cutaneous sensation. We got our cutaneous sensations. We've looked at how we are able to discriminate where touch is coming from with our tactile discrimination. Now we're going to move on to the special senses. In this case, special senses, the first we're going to touch on are taste and olfaction. Okay, smell. And both of these are chemoreceptors. Okay, they're chemoreceptors in that food has chemicals that you put in your mouth. That's what these receptors are going to respond to for taste. And the stuff we breathe in, the air we breathe in, has molecules in it that will elicit responses and receptors in the nose for our sense of smell or olfaction. Okay, these two are interesting in that they are greatly tied together. Our smell will influence our gustation. When you go into, say, grandma's house and she's cooking her famous chocolate chip cookies, you can smell them and you can smell that aroma and you can almost taste it in your mouth. And when you eat it, that just smell makes that sense of taste that much greater. So they are tightly tied together, these two senses. Okay, but we're going to look at how they respond, how we get them to elicit a response. But again, they are both receptors are going to respond to chemicals. So hopefully this slide looks familiar from your anatomy days, but we have taste, and we had those taste buds that are located in those regions on your tongue, and they have these little like onion shaped arrangements with these taste buds with these long gustatory cells in here. Okay, with these little pores and little hairs from the taste cells or gustatory cells sitting in these little pores, in which the chemicals that are coming along the tongue and the saliva can come down and elicit response in the receptors. Okay, these are specialized epithelial, or the cells are here, and they are, you can see each of these cells is going to have a sensory neuron that is associated with it. So when this cell gets activated, it's going to talk to the sensory neuron. If it's strong enough, it's going to send a signal to the sensory neuron and release neurotransmitter to the sensory neuron, and it's going to elicit an action potential in that sensory neuron. Okay. These taste buds, they consist, basically this onion is going to have 50 to 100 specialized of these epithelial cells with these little taste hairs out here in which the receptors are going to reside. Okay. These taste cells, again, they're specialized epithelial, but they behave like neurons. When they get depolarized by activation of the receptors for their different chemicals that are going to activate them, they release the neurotransmitter. The neurotransmitter is released to the sensory neuron, and the sensory neuron has receptors for the neurotransmitter, and they're going to activate the receptor. Okay. Each taste bud, there are cells, there are going to be 5 different tastes, and each taste bud can have cells for those different tastes. Okay. But these cells, see how this is one neuron here, and it's connected to these cells here. These cells would all be of the same taste. They would only respond to, say, sweet is the taste. They would only respond to sweet to elicit a response here. They would have the receptors for sweet on them. Okay, you're not going to have, say, sweet, salty, sour, and they're all eliciting a response on the same neuron, because they would all elicit the same area of the brain. So these cells are specific to one taste, and they will only elicit, this neuron will only be associated with cells of that certain taste. Okay. Other thing we're going to see is taste can be appetitive or aversive. Appetitive meaning when you eat it, you want to eat more. Sweet's usually one of those. You eat some chocolate, you're like, give me some more chocolate, right? If you eat something bitter, though, say you eat something moldy, has a bittery taste, you want to stay away. Okay, and some taste can be appetitive at a certain amount, but aversive when it gets, say, too high in concentration. Okay, salt is one of those. This is from your anatomy days. These are where the papillae, where these taste buds are actually located in. You can see here, folate, the valate, those are all ones in which you can find the little taste buds. All right, I'm not going to test you on this. I just want you to know there is regions that have the taste buds, and this is where those taste buds are going to sit. Okay, I'm more worried or more concerned in that you understand how we elicit a response in these cells of the taste bud. All right, and like I said, there are 5 categories of taste. We got salty, sour, sweet, bitter, and the newest and sometimes called a flavor enhancer is umami, or meaty flavor. Okay, it was once thought that taste, there were certain regions of the tongue that were involved with certain tastes. Now it's believed that all regions that have taste buds can respond to these different tastes. Okay, other things that have an effect on taste is texture and temperature. Okay, temperature. If you're eating something cold, it actually decreases the sweetness of it. So you can imagine stuff that is cold, that is sweet, it has to have a lot of sugar or lots of stuff to elicit a sweet response. Drinking one of those frappuccinos, very, very sweet, but also very cold. So it dampens your sweetness, but you still feel sweet. So you can imagine how much sugar are in these, say, ice cream too, or an ice cream, a popsicle. Okay, gonna dull your sweetness in your taste buds, but you still feel sweet. So you can imagine there is lots of sugar in those colder things that taste sweet because they are influenced by that temperature. All right, so let's look at how each of these receptor cells, these sensory cells, get activated to their respective taste. So again, we have salty, we got sour, we got bitter, we got umami, and we got sweet, right? And there's going to be differing cells with differing receptors that respond to these different chemicals. In the case of salty, what do we consider salty? Salt, sodium, and sodium chloride, right? That makes up our salt. So let's see, foods that are high in sodium chloride would have high sodium ions in them. And what do we see with these? Again, these are specialized epithelium, these receptor cells. Okay, these are the gustatory cells. And in these gustatory cells that respond to salt, give us a salty taste, they have receptors that are channels for sodium. And if there's channels for sodium, if there's salty food, there's lots of sodium, so that sodium's going to enter into the cell, bringing with it its positive charge, depolarizing the cell, and what do you see taking place? Very similar to what you see taking place at the synaptic knobs of our neurons. We have opening of calcium channels, voltage-gated calcium channels, calcium rushes in, and that cues the release of neurotransmitters from these receptor cells. Okay, and we get neurotransmitter release, and if it is enough excitatory signal,
above threshold, that's going to send a signal, start the signal in that neuron. Okay, what we see with salty is salt is actually attractive. You're eating potato chips, these are very good, but at a certain point at higher concentrations, it actually becomes an aversive taste. You don't want too much of it. All right. The next, we got our sour taste. And sour foods are those that have hydrogen ions. They are acidic foods. Okay? Hydrogen ions, what do they have in them? A positive charge. So you eat an acidic food, there's going to be lots of these protons out there. And very similar signaling to the salty taste, in that these cells that respond to sour are going to have channels that allow those protons to enter the cell. And again, same thing happens. Positive charge coming in, that causes depolarization. That causes the opening of those calcium channels, and we get the cue or release of neurotransmitter. Okay. Now, sweet and umami tastes. They activate, we don't have receptor channels here. We have G protein coupled receptors called goose-dukins. Okay, goose-dukins. All right, umami is a meaty flavor. And what we're going to see, umami, the receptors for umami respond to amino acids. What do meat have? It's made of proteins, so there are going to be amino acids. So more amino acids, more meat, it's going to activate these receptors more strongly. Sweet is going to be, any of those sugars are going to activate a sweet G protein couple or goose-dukin. Okay, so these sweet receptors, multi-ligands like fructose, sucrose, or fructose, glucose, lactose, those would all activate these sweet receptors. Umami respond to L-glutamate aspartate, which are present in proteins. So it'd be large amounts. The more protein, the more of this available, and the stronger the signal would be at the goose-dukins. All right, but you'd have cells that would have sugar. You wouldn't have cells that have sugar and the umami receptor there. You would have cells with the sweet receptor, and then you would have other cells that would have the receptor that would respond to those amino acids. Okay, but again, we're activating a G protein, so no channel here. Binding of it activates a G protein. The G proteins are going to activate a second messenger system, and in this case we're going to get closing of potassium channels. Potassium can't leave, that will bring the membrane potential up. We get depolarization, and the depolarization is going to cue the release of neurotransmitter. Okay. We'll find that it actually binding of different molecules can elicit different pathways in these goose-dukins. All right, remember we can go to adenylate cyclase, IP3, those signalings through our G proteins. And that's what's happening here, like sweet. There are molecules that will elicit activation of adenylate cyclase, whereas some molecules may activate that IP3 pathway or calcium channel pathway. Okay, depending on the molecule that's binding can determine what the response may be in the cell. And lastly, we have bitter. Bitter, again, we're going to have a G protein coupled receptor, and it's going to have affinity for many different molecules. Many molecules can bind that give bitter. But what's key on bitter is it is very aversive at low concentration. It's a protective measure. Things that are usually bitter are like spoiled food, so forth. You don't want to be eating these, and so bitter gives you an aversive taste where you don't want to eat them. Okay, but you can see similar to the sweet and umami, second messenger system, it's going to cue calcium release from this endoplasmic reticulum, and that calcium then is going to cue the release of the neurotransmitter. That's how we get our taste. We get our different tastes via these different receptor cells. All right, so once we have that information, or we have that signal sent, we got the transduction, we're sending up, we got the sensory neuron activated, you'll see we get our first order neuron, second order, so it's a 3 neuron pathway for the taste pathways. They're going to flow through these facial or glossopharyngeal, depending on which area is activated for the taste. And you can see brought up, where does that information, all good sensory information, go? We get to the thalamus, and in the thalamus, instead of what we saw with somatosensory sensations that went to the somatosensory cortex, the thalamus is going to slice that information out to that gustatory cortex sitting here, all right, where we can get perception of what taste we are getting. So for this first part, the last sense we'll look at is olfaction, looking at that sense of smell. And in this case, we're going to have olfactory neurons as receptors. So in this case, it is actually a neuron that is going to have the receptor. Okay, the dendrite, the dendritic ends, okay, have these cilias in which the receptors lie, the olfaction receptors that are going to respond. Again, they are chemoreceptors, so these receptors are going to respond to various chemicals that you breathe in through your nose. What we see, there are roughly 380 functional genes that code for different receptors for smell. Okay, this is one of the oldest senses. There are lots of genes put together to sense for smell. One receptor cell, one type of olfactory receptor. So you're not going to have a bunch of many different receptors on the same cell. You're going to have one type of receptor. So you can see we can have many different cells, 380 different types of cells, if each one of those is going to have just one type of receptor on it. Okay. And the signaling of the cell is specific to a specific molecule. All right, so here's the setup of what we call the olfactory epithelium. Right, it sits above the roof of the nasal cavity, and here are what we call the receptor cells, are these bipolar neurons. Right, that's these here, sitting with the little hair projections. This is where you would find the receptors. Okay, and this is a mucus layer. So what happens is we breathe in, the air is running across, the odor molecules are going to dissolve in this mucus and then come and they can bind to the receptors there and activate these bipolar neuron. Okay. The supporting cells, all right, or sustanecular cells, that are these here. Okay, they're basically here to support. These are neurons. Again, they need to have that environment so they can operate. That is basically the job of those supporting cells there. Okay. They oxidize. One of the functions is they oxidize hydrophobic molecules, because hydrophobics would just dissolve right into the membrane. So they limit the permeability, and so those hydrophobic molecules will then elicit a response on a membrane protein. Okay. The other cells in this epithelial are these basal cells. red ones here, and these are, imagine we have damage to these sensory cells, so they have to be replaced. Okay, this is one of the sites where we have neural stem cells, because what are these receptors? They're bipolar neurons. All right, so they get damaged, we get rid of them, and these basal cells can differentiate into new bipolar neurons, or the receptor cells. The other setup, you look at one thing from your anatomy days, should kind of look familiar. You remember that cribriform plate right there in the ethmoid. Okay, if you remember, it has all those little holes, those olfactory foramini, and what do you think goes through there is these axons from the bipolar neurons travel through those holes, those olfactory foramini in the cribriform plate. And what sits on those cribriform plates are the two olfactory bulbs. This is where they're going to synapse with the second order neuron. Okay, how does signaling? Signaling is through, again, the odor molecule will activate a G protein coupled receptor. Right? In this case, the G protein is going to activate adenylate cyclase, and then that cyclic AMP can go in to open up CAMP-gated ion channels, allowing sodium and potassium, and we can get depolarization of the bipolar neuron. If it's strong enough signal, we reach threshold, and now those neurons are firing, and we can send it through the pathway up to the brain to get our perception of what smells are there. Right, so here's kind of that setup again. Here's the bipolar neurons, the axon, and what we see is they synapse with the second order neuron in these spherical arrangements, right? Receive input from one sensory cells. So this would be, these blue would all be activated by the same molecule. They send up, go here, right? Get into the second order neuron, right? The mitral cells, and you get brought up. You'll see these paraglomerular cells going across. Okay, very similar to what we saw with the tactile discrimination, right? These may be similar senses, okay, or similar type of odorants, in that this paraglomerular cell, say we're activating the red here, we can laterally inhibit the blue here, and we can fine point that sensory smell. Okay. We'll see that second order neuron projects to the cerebral cortex. So what is different between this olfactory senses compared to what the other senses that we've seen so far? Our other senses, they went, second order neuron goes to the thalamus, and then the thalamus pieces it out to where it needs to go. Olfactory, only sense that does not go through the thalamus, in that the second order neuron projects directly back to that olfactory cortex. Okay, so no projection to the thalamus. So there's a projection, right? So you're there. And the olfactory cortex is going to get information from signaling. Some of these odor molecules will bind to a high affinity to a certain receptor, but a low affinity to other receptors. So it might have a different smell than one that binds to, say, medium affinity to one receptor and just medium to this other receptor. The brain can interpret these and give you a perception of different smells. Right? So there's lots of information coming up for this to get that sensation of olfaction. All right, so this is going to end for our part 1. In part 2, we'll be getting through, we'll go into looking at what's happening in the ear and what's happening in the eye. We got equilibrium, we got hearing or auditory in our ear, our inner ear, and then we get visual. Okay, the eyes, a long haul. There's a lot of stuff going on, and a lot of has to go right to get our visual information. So we'll go through those in part 2 of the sensory physiology.